Welcome to the physiology of the gastrointestinal tract. Um, this will be the third lecture talking about secretion. Um, you remember that we've divided the topic gastrointestinal physiology into two parts, general physiology and special physiology. In the general physiology, we've talked about the definition of the gut and the structure of the gut and the control systems that orchestrate the different functions of the gut. Motility, absorption, secretion, and digestion. And the second part of the lectures, we started talking about special gastrointestinal physiology. We talked about the special gut functions. We talked about first the motility, and then we spent the past couple lectures uh, talking about the second function of the gastrointestinal tract, which is secretion. In the secretion lecture, we mentioned that the sources of gastrointestinal secretions are four different sources. As a matter of fact, five different sources. The first one is epithelial cells. The second one is salivary glands. The third one is gastric secretion. The fourth one is intestinal secretions, or pancreatic and intestinal secretions. And then the fifth one, which we'll be covering today, is the gallbladder secretions. Uh, Now, we said that the secretions of the epithelial cells consist of water, mucus, and also special molecules. We also mentioned that the salivary glands secrete um, mucus and serous secretions, sometimes individually and sometimes mixed together, depending on the type of salivary gland. We then talked about the gastric secretions, and we said there are four different gastric secretions: the hydrochloric acid, the pepsinogen, the mucus, and then the intrinsic factor, which is important for absorption of vitamin B12. And then we've talked about the pancreatic secretions, which we divided them into endocrine and exocrine secretions. The endocrine secretions were insulin and glucagon, which they have pretty important function in glucose control, in the control of the glucose levels in the blood. And the exocrine pancreatic secretions, we've divided them into two parts. One that's coming from the acinar cells and one that's coming from the ductal cells. The acinar cells basically secrete the digestive enzymes, lipase, amylase, and trypsinogen, whereas the salt and also bicarb are secreted from the ductal cells. And all this is basically vagally and hormonally controlled. The hormone was CCK, or cholecystokinin, which stimulates the secretion of the exocrine pancreatic secretions. And today we will continue on the secretion topic, and we will be talking about the secretions coming from the gallbladder. So, so what is the gallbladder? The gallbladder is a sac-like structure that's attached to the liver, and it secretes uh, gall, which contains bile and other molecules that we will be talking about in few minutes. The contraction of the gallbladder, if you remember when we talked about the gastrointestinal hormones, I talked about the cholecystokinin gut hormone, which the name of cholecystokinin in Latin means the contractor of the gallbladder, and that's where the original name came from in 1928. Now, now, in addition to the hormonal control by CCK on the gallbladder, there's also vagal control on the gallbladder. So basically, if we cut the vagus nerve, we will be inhibiting the contractions of the gallbladder. So that's a very important um, um, concept. we have two types of controls. We have the vagal control and we have the hormonal control via via cholecystokinin or CCK. But next we will be talking about how is the gallbladder secretions are important in the gastrointestinal functions, which is our main topic here. Well, the functions of bile, which is secreted from the gallbladder, is first, it's involved in fat digestion. Fat digestion because it's hydrophobic. Fat is hydrophobic. Bile attaches to fat and gives it an electrical charge, make it hydrophilic, which basically allow digestive enzymes such as lipase and colipase and others to work on fat to digest it. This step, attaching bile to fat droplets, we call that emulsification. Another word for it is saponification, just like how the soap work when we clean our hands. The soap basically attaches to the dirt droplets on our hands and gives them an electrical charge, which makes a chemical reaction, a medium for a chemical reaction, to make it easier to be detached from our skin. That's how we clean our hands, basically, in simple terminology. Now, the digestion of fat is exactly the same. We will talk about that again when we talk about digestion, but for now, please remember that bile attaches to the fat in the gastrointestinal tract and gives it an electrical charge. This process is called emulsification, and this process, the emulsification, is important for digestive enzymes to work on fat, to digest fat, to break fat. Okay? The other function for bile is it's involved in fat absorption through a process called Micelle formation. Micelle formation is basically attaching a bile salt to the very basic components of fat. The very basic components of fat, fatty acids and triglycerides and others, so it can move, it can cross the apical membrane of the enterocyte. So fat or fat digestion product, the end product of fat digestion, will get into the inside of the enterocyte rather than staying in the lumen of the gut. That's the first step in absorption, and that's how. And I'm going to explain it even in more details when I talk about digestion and absorption in the next lecture. This absorption process is called micelle formation. So the digestion process is called emulsification. The fat absorption process is called micelle formation. And also bile is important for elimination of certain products, cholesterol, heavy metals, toxic materials, bile pigments, etc., drugs also, and things like that. Let me show you how in the structure of bile first, and then we'll talk a little bit about how the digestion of fat and the absorption, which I mentioned in the first 2 points, take place. So this is the basic structure of bile acids, basically, which is the major component of bile. We'll talk about the components now of bile, but the major component is bile acids, basically about 50% is bile acids in the bile secretions. So this picture is divided into two. The upper one is the chemical structure of the bile acid, and the bottom one is a a cartoon of how is the bile salt is formed and where are the uh, chemical groups, the carboxyl group basically that gives it an electrical charge, so it can better attach to fat, and then it can work with so the lipase and the colipase start working on it, and the phospholipases and things like that. So this is how it's attaches the carbox group in the top in the chemical structure of the bile salt. In this case, glycolic acid is the bile acid, and the bottom one shows where these carboxyl group are. This is how the carboxyl become um, uh, electrically charged or attractive hydrophilic for the chemical medium. This how the structure of the bile acid is. Now here's the structure that we've seen attaching to fatty droplets in the lumen of the gastrointestinal tract. Once the bile salt it attaches, of course, bile salts. It's not going to be one bile salt by itself. It's going to be multiple bile salts coming with phospholipids, which is another component of bile. Also, we will talk about that in a second. This is in the lumen of the gut. This gives a hydrophilic properties to the fat. Now fat does not become hydrophobic. It starts to become hydrophilic, and that is when the lipase start working on it. When the bile acid or bile salt with the phospholipid attached to the fat droplet, now lipase can act on the fat droplet to digest it and to break it. This process is called emulsification. Again, that's why bisalt is important, and phospholipid, which we will also talk about in a second, will be also important. This is the process of the micelle formation, which is basically important for absorption of fat. Fat droplets cannot enter into the enterocyte in the lumen of the gut and then goes to the bloodstream as is. It needs to be broken. First, we break it. We break it by the previous process that I just talked about, emulsification. That's where bile acids or bile salts attaches to the fat droplet with phospholipids, and lipase starts to work on it. Now, when the digestion process is at the very end, you will see that the bile salt is basically attaching to the fatty acids, the basic fatty acids, and it basically transported to the apical membrane or apical membrane of the enterocyte. That's how it helps these fatty acids to get inside the cell from the lumen of the gut into inside of the cell where the shaded arrow is pointing from the lumen toward the inside of the cell. That's how the absorption takes place. And without the bile acid, this process is not going to happen. So, and this process is called micelle formation. So that without this process, there's no fat absorption, and without the previous process, there's no fat digestion. So bile acids are very, very, very crucial in both processes, the digestion of fat and the absorption of fat. So what are the components of bile? There are a number of components in bile. So we have organic component, and we have inorganic components. The organic component, just like saliva has organic component and inorganic component. The organic component in bile, or in bile secretions, if you want, there are four. There are bile acids, phospholipids, cholesterol and bile pigment, bile acids, phospholipids, cholesterol, and bile pigments. Cholesterol and bile pigments, as a matter of fact, as I'll talk about later, they will become, if they are increasing in amount, if their amount is increased, they become problematic in causing gallstones, and that's an important thing clinically. Inorganic component of bile consists of water, bicarbs, and different ions. So let's take the first component, the bile acids, first, and talk about it and see what's in there. We have a total of about 2.5 grams of bile secretions. 600 milligram are synthesized daily from the bile secretions, from all organic and inorganic, all of the components, all 4 or 5 components, all organic and inorganic components. They're 2.5 grams. 600 milligrams of this 2.5 grams are synthesized daily. The remaining, is recycled, basically. More than 50% of bile secretions are bile acids. So half of bile secretions are basically bile acids, which basically explains why bile is extremely important for the digestion and absorption of fat, because of the 2 processes that I mentioned earlier, emulsification and Micelle formation. Very important. So without bile, there's no absorption, and there's no digestion, and there's no absorption of fat. Bile secretions are synthesized from cholesterol. Cholesterol is the main nucleus for all bile secretions. All bile secretions, they will have cholesterol molecule in them, and 90 to 95% of bile secretions are bound to plasma protein. They're not going to be flying by themselves. They're not going to be flying solo, as they say. They will be always attached to plasma proteins. Let's see how these are some examples that I'm going to show you here. So you have primary bile acids, you have secondary bile acids, and you have bile salts. Primary, secondary, and bile salts. You have cholic acid, you have deoxycholic acid. This is a primary, and this is a secondary. All of these are examples. And the very bottom of the picture shows you where the cholesterol is. Lecithin, which is a phospholipid also. Taurine, again, a bile salt. All of these, glycine, all of these are basically different structures of different bile acids, basically. Primary and secondary. Primary and secondary. Now, where are the first component, or the bile acids, which makes 50%? As I mentioned, the components are organic and inorganic. The organic are bile acids, phospholipids, cholesterol, and bile pigment. Inorganic, water, bicarbs, and iron. Now we're going to talk about the very first organic component, and that is the bile acids. Oh, I meant to mention that also the primary ones are synthesized in the liver, whereas the secondary ones, they're synthesized by bacteria. Okay, so now bile acids. We choose to talk about bile acids first because it forms half of bile secretions. 50% of your bile secretions consist of bile acids. They're basically synthesized in the hepatocytes or in the liver, okay, and they're continuously produced. Why? Because there's always food in the gut. There's always food in the gut, and that's why they're always and continuously produced. They are secreted, however, 30 minutes after the meal, immediately, or I shouldn't say immediately, 30 minutes after a meal, the bile acids start to be secreted. And as I mentioned earlier, the bile acid are under the control or of, of the hormone cholecystokinin and the vagus nerve. So if you take so you cut the vagus, you inhibit the production of bile acids, and when you take CCK away, or if you give a CCK antagonist, you will have inhibition of uh, bile acid production and control and contraction of the gall bladder. So what are the main characteristics of these bile acids? I mentioned earlier that they're always conjugated. Bacteria unconjugate them for a while, and then 50% of them are passively reabsorbed, especially from the upper intestine, and they are unconjugated, and 50% are actively reabsorbed from the lower intestine, and they are conjugated. So when you collect bile acids, you will have them all conjugated, despite the fact that they are reabsorbed. Half of them is reabsorbed from the upper intestine, and half of them is reabsorbed from the lower intestine. It's as simple as that. Okay? Unconjugated and conjugated, at the very end point, they are all conjugated. So this is a very important thing to remember, that 50% are absorbed from the upper gut, 50% are reabsorbed from the lower gut. And this diagram basically explains what I've just talked about. The liver produces bile. Bile gets into the duodenum. Of course, you utilize everything for the digestion of fat, all the way up to 95%. Bile acids are reabsorbed through the portal blood circulation back to the liver. 55% of them, 5% is going with the feces, and that's why we said 90 to 95% basically are reused. That's very important, and here we have to remember that cholecystokinin and the vagus, which is represented by ACH, which is acetylcholine, the parasympathetic neurotransmitter in the vagus, both of them help in the contraction of the gallbladder to release bile, to secrete bile. If you cut the vagus, or if you block CCK with a CCK1 receptor or CCKA receptor antagonist, what you will have is inhibition of this contraction. So the gallbladder will not be able to contract, and as a result, bile will not be secreted into the duodenum. That's the extent of the bile acids. 50% of your bile secretions is basically bile acids. Now, the second component or organic component of bile secretions is the second component of bile secretion is the phospholipids. It's about 30 to 40%. So bile acids is about 50%. This one is about 30 to 40%. It's about a total of 90%. And lecithin, which is also basically amphipathic, which means that it is electrically charged. It is basically important for making the lipase work on them, and I explained that when I said emulsification is the process for that. when the lecithin, which is a phospholipid, attaches to the fat droplet along with the bile acid in the slide that I've talked about, the emulsification, the molecule, the whole molecule becomes amphipathic, becomes uh, attracting to lipase to work on it, hydrophilic. It likes water. It likes the reaction, and so that's how the micelle formation for the absorption of fat also is taking place. So emulsification as well as micelle formation. Uh, okay, um, now we will talk about the cholesterol, which is the third component of the organic biliary secretions. Cholesterol accounts for about 4% of the total bile secretions. Remember, 50% is bile acids or bile salts. 40% is phospholipids, lecithin and things like that, and then 4% is cholesterol. And the most important thing about cholesterol, it is important, and it works for everything in the fat metabolism. So it's a very important molecule. But the bad part about it is that any excess of cholesterol may serve as a nidus for gallstone formation. Any excess of cholesterol may act as a nidus for gallstone formation in the gallbladder. Now, the last component of the organic uh, uh, part of the biliary secretions or bile is the bile pigments. Bile pigment account for about 2%, and it comes from the the old RBCs when they are destroyed in the liver, and or during liver damage during a disease. Bilirubin is one of the most important bile pigments, and it'll be conjugated to glucuronic acid in the liver. So if it's not there, that means we have jaundice or one of the diseases that is manifested by yellowish mucous membranes or yellowish sclera in the eye. So this is a very important molecule, if you will, or component of bile secretions, the bilirubin. And again, it's the bile pigment, the excessive amount of it will also, just like cholesterol, will serve as a nidus for gallstones, just like cholesterol that I mentioned a second ago. Bile pigment will not be found in the micelle, so it's only bile acids that or bile salts will be found in the micelle formation, which basically help, as I mentioned earlier, in the absorption of fat, in crossing fat molecule from the lumen of the gut to the inside of the enterocyte. That's a very important thing to remember. They don't get into the enterocyte. The micelle does not get into the enterocyte. The bile pigment is not present in the micelle. The only thing that's present in the micelle is from the bile secretions, is only the bile salt or bile acid. Same thing, bile salt or bile acid. It's a carrier for the very basic components of fat, which is fatty acids. It carries them all the way to the membrane of the enterocyte. After that, it leaves them there, and it goes back to be reabsorbed to the liver. It does not get into the enterocyte. It does not get into the enterocyte. The micelle does not The bile salt does not get into the enterocyte. The only thing is the basic component of fat digestion, which is fatty acids or triglycerides or things like cholesterol or things like that. This is a picture that shows uh, the stones in the gallbladder, and again it's attached to the liver, and when it blocks the the basically the duct that releases that have the bile going to the upper small intestine, it's a problem in fat digestion. So fat digestion basically gets compromised, and also there's abdominal pain, severe abdominal pain that's associated with it, and things like that, which requires surgery, as we will talk about in a second. Gallstones, I mentioned that before. There are two types, either cholesterol-based or pigment-based, bilirubin based. Most of them, however, are 50 to 75% are cholesterol. Most of the gallstones are basically cholesterol based. Um, this is very important, and um this requires surgery, removal of the gallbladder, which we will refer to as cholecystectomy. Cholecystectomy, removal of the gallbladder. This will not affect the digestion of fat, by the way, because you will have sources of lipases coming from the the pancreas, as we remember from the acinar cells, and also you have bile that will be secreted directly from the liver to the intestines based on the meal consumption. So that's an important thing to remember. It will not taking the gallbladder out in surgery will not affect digestion. Of course, it requires diet modifications immediately after surgery, but after that things will go smoother in the postoperative time, about 3 to 6 months in general. Uh, the last component of bile secretions is the inorganic component, and that is it consists of water, bicarbs, and iron, and different ions, I mean, and all of them are basically present. They help in concentrating the bile to become extremely concentrated once they are um, absorbed. Um, Now this will mark the end of our uh, discussion on the second function of the gastrointestinal tract, and that is secretions. We've talked about secretions coming from 5 different sources: epithelial cells, salivary glands, stomach, pancreas, and gallbladder. 5 of them. In the next lecture, I will start on another special function of the gastrointestinal tract, and that is digestion and absorption. I will combine digestion and absorption in the same lecture. So you will see how you digest the food and then you absorb it, how the mechanism is complementary, how it's going to be easier to understand rather than to divide them into 2 different